Thank you for the introduction as well. And good morning, everyone. So I thought I'd start by just kind of introducing myself to the audience for anyone who doesn't know the research that I do and sort of how I was trained, but this little diagram will help. So I did my PhD and postdoc studying eye movement control, but really being trained in monkey neurophysiology, where we would lower microelectrodes in the brain and record from single neurons and correlate the activity of neurons to behavior. And way back now, several decades ago, when I first got to Queens, it was my intention to rely on translation and to use this wealth of data we were collecting from monkeys to help interpret and understand human behavior if the humans were doing the exact same behavioral tasks as the monkeys. And so we studied healthy human behavior, and we've studied many different neurological psychiatric disease conditions. And this is a lot of what I'm going to talk about today. There's another whole arm of work in my lab that's been started only in the last decade or so, where we're translating back, or this reverse translation of trying to establish elements of human disease conditions in the monkey to see how they perturb the biomarkers that we study. But I'm not going to focus today on this arm of my research. So in terms of an outline, I thought I'd try to hit on 4 main points today. Is to introduce you as the audience to the circuitry in the brain and our extensive knowledge of the circuitry that's used to control saccadic eye movements. And then I'm going to elaborate on that and move towards pupil control as well. I'm going to anchor this with some key monkey behavioral neurophysiology findings that are critical to then use this neurophysiology to interpret the data that we get from humans doing various eye tracking experiments. And I'll focus on 2 parts here today. First of all, talking about normal development and aging and how saccade, pupil, and blink behavior changes across the lifespan, and how we can use this same approach to identify biomarkers of disease. And I'll focus today on neurodegenerative diseases. So why is the eye movement system so attractive to study? And one of the reasons is it spans almost the entire neuraxis. So it's not that the whole brain is involved in controlling eye movements, but if you consider the brain areas that are involved in vision and those that are involved in some way selecting how to move your eyes to facilitate visual processing, there are areas over the entire neuraxis except the spinal cord. And what this means is if you set up the right kind of experimental condition or paradigm, you should be able to identify abnormalities in some kind of way if there's any problems in the brain, because this circuitry is so extensive. And just to highlight how extensive this circuitry is, I've got an oversimplified version here, and I stress the word oversimplified. And I've really only tried to highlight some of the core elements here, and I can examine you on all the acronyms after that are defined over here. I actually use this slide to show my colleagues that are molecular biologists when they show their intimidating signaling pathways. I come back with this. But for the sake of today, what's going to be important is the color in this figure, not the names of the little boxes. But this is extensive circuitry all through the cortex, the basal ganglia, the brainstem. And these circuits ultimately control saccadic eye movements, blink behavior, pupil constriction and dilation. And all of these behaviors we can capture with a single video camera that's remote to an individual's face, just in front and down below. And we can get an image and use that to extrapolate eye movements, blink behavior, and pupil size and what they might mean. And over the many years of my career now, we've been putting microelectrodes in different areas of the brain here to understand this circuit And a key brain area in my world here, right in the center, is the superior colliculus, where I've spent more time recording there than anywhere else in the brain. And I might have spent more time recording there than just about anywhere else in the world now, which is a bit scary. But there's a number of other places in this circuit where we've gone and recorded in different experiments in awake monkeys doing various eye tracking experiments. And so, we've been across a large part of the neuraxis here and have a good understanding of it. Now, this circuit wasn't just built up by our lab. This is the entire ocular motor field contributed to this, but we've played an active role in building that knowledge as well. So in our lab, the key part to do this translation between the monkey behavioral neurophysiology and the human clinical work is that we use a set of behavioral tasks that are transferable between the two. So what I mean by that is all of the tasks that I'm going to show you today, we have monkeys trained to perform those exact same tasks that we ask the humans to perform. And that's absolutely critical so we can use the monkey neurophysiology to interpret the human behavior. So I'm going to focus today on a set of structured tasks here, which is an interleaved pro-saccade and anti-saccade task. And I'll get into the details of this a lot more in my talk. I won't talk today about this free viewing unstructured task, but this is rapidly overtaking our lab as becoming the dominant tool going forward. Okay, so what I'm going to do is start to introduce now some key neurophysiological concepts that'll be critical to understand the human behavior when I get there. So this is now the simplified circuit. So now we can pay attention to these brain areas. And the key about the superior colliculus, the intermediate layers here, is this convergence of input from early visual system, parietal cortex, frontal cortex, not shown on here, temporal cortex, the basal ganglia, and a host of other subcortical regions. So this is really a hub of integration. And in the point where, with the point of saccadic eye movements, this is really motor cortex for making saccades. This is the command structure. The intermediate colliculus, the commands go to the premotor circuit, the behavior gets executed. So I'm going to just open this box up a little more and show you what's inside. There's these key circuits down to the brainstem reticular formation. I won't go into the details of this. But the superior colliculus has a map, an oculomotor map, a retinotopic map of eye movement space. And the neurons on this map then project to these brainstem premotor circuits to send the command signals to control behavior. And we've been recording for years and years across this map, and I identified a number of different signals. And I want to highlight only two contrasting signals here for today. One we call a saccade neuron. So this has been well identified in the literature. These are neurons that generate a very high frequency burst of action potentials here when the monkey makes a change in eye position that's a rapid saccadic eye movement. And we identified that this map extrapolates to a zero position. And so up in the rostral lateral pole of the superior colliculus are neurons that we called them fixation neurons because they're kind of the opposite. They're tonically active when the eye is fixed, and there's a pause in discharge for this saccade when this saccade command is coming through. So these two neuron types are kind of like reciprocal in their discharge. They both project to the brainstem. These reciprocal patterns have identified down in the brainstem as well. And these reciprocal patterns have been identified throughout the basal ganglia and the frontal cortex. So this is not unique to the superior colliculus, but the colliculus is the place where we have the window into this with the map, so we can really make sense of these signals. Okay, so the first task I want to talk to you about is we call a pro-saccade task. Imagine you're the subject. You're looking at a spot of light on the screen. It disappears for a couple hundred milliseconds. and then randomly a white target will appear on the left or the right side, and your job is just look at the target. So this is very simple behavior, automatic response, high stimulus response compatibility. If you were doing a block of these trials, you don't really need to think about what you're doing. Your thoughts could drift into what you're going to make for dinner tonight or what you're going to watch on TV tonight, and you'd still generate perfectly normal behavior while you're generating this task. But I want to show you a couple of key things of what we identified in the monkey superior colliculus here. So first of all, a generation ago, Fisher and Bach identified this distribution of saccadic reaction times here. This is supposed to say saccade reaction time here. Time 0 right here is when the target comes on, and then we get some distribution of saccades. And he identified this early peak that he called express saccades, and then a later peak he called regular saccades, kind of akin to if you're sending a letter in the mail, you can send it by express post or regular post. And the express saccades come out here really fast, 90 or 100 milliseconds. This is now the recording of one of these saccade neurons in the superior colliculus. So these are a number of trials here. The eye position trace is superimposed. This is a raster plot of each line here is a separate trial, and each tick is an action potential from this one saccade neuron. And then those action potentials are summed together and smoothed out a bit to produce this spike density waveform, which is the important trace to follow in all of my neurophysiology figures. And you can see here when the monkey generated these regular latency saccades, this cell had 2 bursts of action potentials. This first burst was time locked to the onset of the peripheral stimulus, which was going to be the saccade target. The second burst here is time locked to the occurrence of the saccade. So this is a sensory response followed by a motor response and the motor command to move the eyes to that sensory stimulus. And the monkey occasionally generated these express saccades, and you can see the latency of these express saccades much shorter. And what's intriguing when you look at the responses of this neuron for the express saccade, there's only one burst now. The same burst is equally well aligned with the onset of the stimulus and the onset of the saccade. So this is a motor burst now. It's sensory being triggered, but it's motor in its execution. So if we look at this and blow this up in a little more detail, and I highlighted under here this yellow strip that I'm going to make a big part of. So the stimulus comes on at this vertical line, and for a regular saccade we get a visual burst and a motor burst, and for the express saccade we get just one big burst. But you'll notice the buildup of activity leading up to that burst was elevated on the trials where we got the express saccades. So if you think about this for a moment, this is pretty cool. If you were in the lab with us when we recorded from this neuron, we can tell you at this moment in time what the monkey's going to do, if he's going to make an express saccade or a regular latency saccade based on the excitability of this neuron. And if this buildup is high enough, it's going to take the system up close to a threshold so that that visual burst will exceed the saccade threshold. And that does not happen for the regular latency saccades. Okay, now in the most of the data I'm going to show you today, we had this pro-saccade task going, but it was interleaved with another task we call the anti-saccade task, where here the fixation light is a different color. In this case, it's red. And it conveys a very different instruction to the animal now, or to the human, so that when the stimulus comes on, now you must look in the opposite direction. This task is really hard to do because of the stimulus response incompatibility. And there are extra stages of processing here, and I'll break them down into really simplified into 2 main steps. First, don't do the automatic response. Don't look at the light. So step 1 is just don't do that. And then step 2 is you have to transform or invert the target vector. And all that means is if we look at this trial here with the stimulus appearing on the left, that means the visual information went into the right hemisphere, but the monkey's required to make a rightward saccade that has to come out of the left hemisphere of the brain. And so we've got all the information's got to cross the midline as well. So now we're using a task that's really engaging the entire nervous system. And we randomly interleave these trials, and we use the color of the fixation spot to convey the instruction. So this is the behavior of monkeys. And if we just look down at the bottom, this is from a block of trials where it was a red fixation spot. That meant make an anti-saccade. The stimulus appeared on the left side of the screen, which meant the monkey was supposed to look to the right side of the screen. So the red traces here represent the correct trials that the monkey got rewarded on. But on many trials, he made a mistake and produced the blue traces where he erroneously looked at the stimulus before he looked away. And this pattern of eye movements is really important for us to see because these are the errors where he looked at the stimulus. But that monkey understood exactly what he was required to do because he corrected those errors within 100 or 200 milliseconds. He knew he had to get his eye over to the right side to get the reward. He just couldn't suppress this reflexive automatic urge to look at the light sometimes. And we're going to see humans do the same thing. Okay, so how does the monkey do this on the anti-saccade trial? Well, we trained monkeys to do this. We trained them for 6 months, and finally they successfully learned how to make anti-saccades. So what changed in the brain? So if we just focus on this one superior colliculus saccade neuron now, and there's 2 traces here. I'm showing you the response of this saccade neuron in blue for pro-saccade trials and in red for anti-saccade trials. And I'm only showing you the trials where the stimulus is in the response field of the neuron. So for the pro-saccade task, the monkey's going to look into the response field of the neuron. For the anti-saccade trials, the monkey has to look in the opposite direction. So 6 months of training, what was going to change in the monkey's brain? And you can see the visual response to that stimulus is virtually identical in the pro-saccade and the anti-saccade trials. And that's probably good. All the training did not change the incoming visual information to the monkey's brain. That would really mess up the rest of his behavior in everything else he had to do. What did change, it looked like, was the baseline activity that visual response was riding on. And so on the pro-saccade trials, this cell had more activity and more buildup during the gap before the stimulus came on than on anti-saccade trials. On anti-saccade trials, it looks like this cell's just being clamped off so that this visual response won't cross the threshold to trigger a saccade. Now, if we flip the data around, now I'm going to show you the trials where the saccade goes in the response field, which means on the pro-saccade trials, the stimulus was in the response field, but on the anti-saccade trials, the stimulus was on the opposite side. So this is the same blue curve on the right and left side, but now the red curve are the trials where the stimulus was on the right and the saccade went to the left. And you can see the result of that was actual inhibition of this cell at the time of the visual response. And then the brain's trying to generate a saccade burst from this neuron to trigger this voluntary anti-saccade, which is going away from the stimulus. And at the same time, on the side of the brain that got the visual response, this side of the brain's trying to be shut down. And so there's a battle between these 2 red curves on opposite sides of the brain, trying to generate the correct anti-saccade, or if this response gets too high here, it will trigger an error on the anti-saccade trials. Okay, so who's doing this? What is, after all these months of training of the monkeys, what's turning down this response? And the result is, if we look at the fixation neurons I already introduced you to, that have this high tonic activity and pause for saccades, and just if we look during the period of fixation here, the fixation neurons in the frontal cortex and in the superior colliculus have more tonic activity on the anti-saccade trials than on the pro-saccade trials. It's the opposite of what was going on with the saccade neurons. So these fixation neurons are then contributing this top-down inhibitory signal onto the saccade neurons to not let this visual response cross the threshold. Okay, the last piece of monkey neurophysiology I want to show you is now when the monkey made a mistake. So this is the distribution of the time of the correct anti-saccades. And this is the distribution of the timing of the errors. Now, this is an overtrained monkey. So most of the errors fall around 90 or 100 milliseconds. They're almost all express saccades. Some of them are longer latency. In the humans we study that are naive, we get a lot more of these longer latency errors. And now if we look over here at what happened with our saccade neuron in the superior colliculus, in red is the correct response of the monkey on anti-saccade trials, the visual response, and the cell gets shut down. And the orange or the salmon trace here is what happened when the monkey made a mistake and looked directly at the stimulus, when the visual response became the motor command and triggered the saccade. And what you can see in the yellow box, the cell is already telling us when the direction error is coming. So if you were in the lab with us the day we recorded from this neuron, we can tell you at this moment in time, before the stimulus has even came on, we can tell you with reasonable certainty what's going to be the outcome, if the monkey's going to make a mistake or not. And it's dependent on how good this top-down inhibition is. Okay, so that was a bit of a crash course in some key observations from monkey neurophysiology that I just wanted to share with you before I go on, then to now show how we're going to use this in studying humans. First, normal human behavior, development and aging, and then what happens in neurodegenerative diseases. So we have a paper literally that just came out last month in Frontiers in Aging, which describes interleaved pro and anti-saccade behavior across the lifespan. I've had the good fortune over my career to maybe publish 200 research papers now. This one has the most person hours invested of any study I've been engaged in. When you look at that, this contains the data of 604 healthy control participants between the ages of 5 and 93, an extensive set of data. And this is our normative control data upon which we use to then compare with all the different clinical studies we're engaged in. And the person who really led this over the finish line is Rachel Yap, a senior PhD student in my lab. So these are now human participants doing the exact same experiment the monkeys did, this interleaved pro and anti-saccade task. And I'm going to show you the behavior across the lifespan, but I'm going to do it in a certain way. So you really need to understand the layout of this slide. And if you get the layout of this, you'll be able to follow the rest of my talk super easy. So what's shown on the left is the pro-saccade behavior. On the right is the anti-saccade behavior. And this is a cumulative distribution here of the correct pro-saccade trials and the direction errors. This happens to be from a group of children between 5 and 8 years of age. There's 29 children in here. There's thousands and thousands of saccades. And this is the distribution of their reaction times when they make pro-saccades. You know, they go from 90 milliseconds out to about 250 or 300 milliseconds. And this gray bar is the region of express saccades that I introduced you before with the monkey neurophysiology. So that's the pro-saccade behavior. They look pretty good. But over here in the anti-saccade task, these kids are horrible. They're making 60% errors, only 40% correct responses. So this is worse than chance. And I hope you can see that this represents a horse race where the direction errors are getting triggered much faster here in the express epoch and belong and beyond. And so they occur much faster than the correct responses, just like in the monkeys. Now we look at some children 9 to 10 years of age, so no difference of behavior really in the pro-saccade task. In the anti-saccade task, we see a significant improvement, less errors, more correct responses. The timing of the correct anti-saccades is getting more robust. Here's 11 to 12-year-olds, even less errors, more correct responses, more automated, or the anti-saccades being generated at more consistent reaction times. You'll notice over here, virtually no difference across these ages in pro-saccade behavior. Here's 13 to 14 years of age, less errors, more correct responses. 15 to 16, now we got a lot of correct responses, good ability to suppress these errors. This is when we consider as a society granting the keys to automobiles to individuals. As we can go on now to the young 20-somethings, they make the least errors, the most correct responses. Now we give them access to alcohol, they have voting rights, etc. So we see this dramatic improvement in the anti-saccade task as their cognitive performance improved. And you can see virtually no difference across those age groups in the pro-saccade task, even though these trials were randomly interleaved. So this dramatic difference is not because of motivation or waning attention or any of that. Their pro-saccade performance shows us the parts of the brain they need to generate pro-saccades are functioning normally. It's the parts required to generate anti-saccades that have big problems early in development. Okay, so here's the same curve, the 21 to 25-year-olds, and now it'll become clear why we call them the Olympic athletes in the anti-saccade task. Here's 25 to 35, 35 to 45, not bad. Into the 50s, 60s, 70s, the 80s. It's not as dramatic as it was in development, but it's starting to fall apart. We're starting to make more errors, and the anti-saccades are getting slower. And we're also getting a bit slower in the pro-saccade task. Okay, so if I put all that together, this slide is only showing pro-saccade performance. This is showing all of the age groups that went from age 5 to the early 20s. And then this is for the aging cohorts that went from the early 20s into the mid 80s. And you can see here with the children, no change in behavior in the pro-saccade task at all. We can see this slowing down as we age, especially over 55 years of age. Same layout now, but now it's the anti-saccade behavior I'm showing you. And you can see in the child development, just dramatic changes in behavior from ages 5 to 25. And we also have big changes in behavior as we go from the 20s to the 80s. So there's a dramatic decrease in these direction errors in both this early gray epoch, which is when there's express saccades, and the longer latency, the regular latency direction errors. They're both changing dramatically. You can see here with the old people, it's more the regular latency errors that are coming out. And this increase with reaction time. This shows now how we fit a model to this. So this is log transformed error rates here. So it's a bit arbitrary what these units are. But as this number goes down, it means less and less errors. This is showing the express saccade error rate as a function of age of all of our subjects. And these are the regular latency errors in the anti-saccade task. So you see both the express errors and the regular latency errors are being reduced dramatically through development up to about age 20 or 20, between 20 and 25. And then after that, we don't get an increase in these early express saccade errors. But as we age, it's the longer latency errors that are starting to come out. And this will be important as we start to look at the patients with neurodegenerative disease. What's interesting here with these express errors, if these jump out, it's more indicative of psychiatric illness. And these longer latency errors are going to be more indicative of neurodegenerative diseases. Okay, so now I want to show you how we can use this to study disease. So over the years in my lab, we have studied many neurological psychiatric diseases. As I got started, I thought we were spreading ourselves so thin here. But then one day, this was maybe 20 years ago, actually, the penny dropped. And in no disrespect to any of the patients, but if I invoke the model of a knockout mouse, effectively, that's the opportunity we have here. That the more of these different kind of patients that we study in the exact same task where we understand the circuit so well, these patients effectively we can be used as knockouts, and we learn more about the basic science of our circuit when we compare the biomarkers that we get across these diseases. Now, of course, in the remaining few minutes of my talk, I can't do justice with that. So I'm only going to focus on a few here, the color-coded diseases here that are all related to neurodegenerative disease. And there's a simple reason why I'm going to focus on all of them, because we targeted all of those patient groups in a single study. So I had the great fortune in my career about 10 years ago to be able to join this pan-Ontario study that was funded by the Ontario Brain Institute called the Ontario Neurodegenerative Disease Research Initiative. The initial leader of this program was actually Mike Strong, who led it the first 5 years. And then 5 years ago, I picked up as a co-lead of this with two neurologists from Sunnybrook, Mario Masalis and Rick Schwartz. And effectively what this study is, we recruited a total of 520 patients with different neurodegenerative diseases. Every one of those patients underwent this incredibly extensive workup that included a full clinical assessment, a blood draw for genetics analysis, 4 hours of neuropsych testing, gait and balance analysis, eye tracking, 6 kinds of neuroimaging, structural and functional imaging, and even retinal imaging. And these patients were followed for 3 years. I'm going to focus right now mostly on just the eye tracking data that emerged at the cross-sectional baseline, but how we're now trying to link it to what data we got from some of these other platforms. And this is the work of 2 senior PhD students in my lab, Heidi Reich and Jeff Wang. So I want to show you now how the behavior of these patients changed in the pro and anti-saccade task. And so this is the distribution of reaction times in the pro-saccade task again, and in the anti-saccade. The positive curves are the correct trials. The negative curves are the reaction times of the errors. And this is the control cohort. These are elderly individuals from 55 to 85 years of age. There's 137 of them there. They do really good in the pro-saccade. They make some errors in the anti-saccade, both express and regular latency errors, but they're pretty good. This now shows the 2 blue lines we separate the cohort we recruited with Alzheimer's disease A third of those patients had full-blown Alzheimer's disease, but 2/3 of them had the prodromal condition of mild cognitive impairment. So many of these MCI patients are going to go on and convert to Alzheimer's disease. We can see in the pro-saccade task not much difference in the behavior compared to the controls. In the anti-saccade task, we see impairments and they appear to be graded. So the dark blue is the MCI condition, the prodromal condition to Alzheimer's disease, which is in lighter blue. And we can see the Alzheimer's patients make more errors in the anti-saccade task, fewer correct responses, more guesses. So their performance is deteriorating from MCI to Alzheimer's on the anti-saccade task, but not much different on the pro-saccade task. And I'll actually point out, you'll notice the two blue lines are running to the left of the gray dashed line. So if anything, the controls are faster in the pro-saccade task. Doesn't usually reach significance, but we consistently find this. This is the group of Parkinson's patients now. And we've divided them into 3 subgroups of Parkinson's based on the relative amount of cognitive impairment. So about 1/3 of the patients, or just over 1/3, maybe about 40%, were classified as cognitively normal based on that 4-hour neuropsych assessment. And then about 1/3 of them were identified as having a mild cognitive impairment, and the other 1/3 having dementia. And when we look at their performance again in the pro-saccade and anti-saccade task, we see really no difference in the pro-saccade task again, other than all these sub-cohorts of Parkinson's, again, running to the left of the controls here in the pro-saccade task, so that they're actually faster than the controls. And then in the anti-saccade task, we pick up the big cognitive impairment. We can see that the cognitively normal in MCI make about 40% errors, and the patients with dementia have many more errors. And when we look at the anti-saccade reaction times, we can see the cognitively normal being faster than the cognitively impaired. So we can very nicely separate out these cohorts and identify their cognitive impairment based on the anti-saccade task. And this is another cohort I won't even bother to identify here now. So these are the different cohorts that were in Andre. And what I'm plotting here is the percentage of these express latency errors. And the horizontal dashed line is where the control values are. And the horizontal bar and all the dots are where all the patients were relative to the mean of the controls. And at the top here, I put asterisks next to the subsets that actually made more direction errors at express latency here compared to controls. And it's only a couple of the subgroups. This is the same plot now, but for the regular latency anti-saccade errors. So again, the horizontal bar is where the controls are. And I hope you can see now these colored dots are all moving up from where the controls are. And now most of these lanes are significantly impaired relative to the controls, with the only exception of amyotrophic lateral sclerosis, which were cognitively the most intact of all the patients we had based on neuropsych testing, and we could see that with their anti-saccade performance. Okay, so now we've got so many of these parameters. I could show you here about 20 of these curves of different pro-saccade or anti-saccade measures that are either normal or abnormal. And we start to lose the forest for the trees. So because we had so much data here, we could employ a factor analysis where we identified the 12 most important measures from pro-saccade and anti-saccade, like reaction time, error rates, velocity or amplitude of the saccades, and a few others here. And when we did a factor analysis, because we had hundreds and hundreds of subjects here, the factor analysis revealed four main factors that were largely independent. And I want to break down how these aligned in the anti-saccade task, because it was absolutely remarkable. Because they dissociated in time in the task, these four factors. And so we have at the start of the trial, the fixation light comes on, and it's either red or green to convey the instruction for pro-saccade in green or anti in red. That lasts for about a second. There's a gap of no stimuli, then the stimulus comes on, and then you're supposed to make your correct pro or anti-saccade. Factor one loaded all the parameters that were measuring things out here related to early fixation. Factor two collapsed all the measures that were around what do you do when the stimulus came on. Factor three loaded more voluntary parameters related to the anti-saccade. And factor four was more motor related to the execution of the saccade. So this first factor was early engagement and disengagement of the task. This second factor was all about preparing for the stimulus coming on and having the appropriate amount of top-down inhibition conveyed by those fixation neurons so that this visual transient doesn't go through the superior colliculus. Factor three was then related to the competition between the automated and voluntary command to trigger ultimately the voluntary saccade to where there was no visual stimulus. And as I said, factor four was just related to the metrics of the saccade. So we were intrigued how these four factors segregated. And so we had the opportunity to then try to correlate these four factors with the main neuropsychological cognitive domain scores that we could extract from those hours of neuropsych testing. So we had a cognitive domain score for attention working memory, executive function, memory, visual spatial function, and language. And we actually went after language because there's language deficits in a lot of the subgroups of neurodegenerative patients we were studying. And we could see which of these cognitive domain scores correlated with the factors that we were studying in the anti-saccade task. And the really important ones that are colored dark, that are the strongest, were really factor one, factor two, and factor three. And they were correlating more with attention working memory, executive function, and visuospatial function, the kind of functions you would expect that you need to properly complete the anti-saccade task. And this factor three had the most of these cognitive domain scores correlated with it. And you can see that this pattern of factor three was very different from factor two. And so we really believe we're on two completely different processes of the brain here, this factor two versus factor three, even though both of them are playing out right in the superior colliculus. But we know how these play out with different neural signals that we were able to measure in the monkeys doing the anti-saccade task. Okay, in the last few minutes, I'm going to start to build on this approach now. And I've added a couple more boxes, incredibly oversimplified here, but linking in the pupil circuit, because we also can get pupil movements for free with our video-based eye tracker. The pupil, we know, is driven by signals that come off the retina of luminance. We've known that for a long time. We've also known for a long time that arousal signals can drive the pupil, and the sympathetic nervous system and its linkage to the locus coeruleus and through limbic cortex provide important arousal signals to the pupil. And more recently, it's been identified in the literature that bottom-up sensory signals can have a correlate on the pupil, as well as top-down attention signals that might come from frontal cortex. This gentleman on the left here, Josh Wang, was a postdoc in my lab for about 10 years. He's now gone back to Taiwan in a faculty position. But his postdoc project was devoted to understanding the linkage between the intermediate colliculus and the pupil premotor circuit. So he basically studied this line right here. And this line's really important because it can deliver all the bottom-up visual signals and the top-down attention signals to the pupil. And that's what Josh embarked on, finding how these signals were coming down through the colliculus to get to the pupil. And Jeff Wang, a senior PhD student in my lab, is now showing how the pupil responses are abnormal in neurodegenerative diseases. So we kind of lucked out here because we started doing this anti-saccade, pro-saccade task before we got on to the pupil recordings. And it turned out that the structure of our paradigm was ideal for revealing a number of pupil measures. So the fixation point, this is time in the task. The fixation point comes on and to signal the pro or anti-saccade task. And then it goes out and there's a gap, and then the target stimulus comes on. And then over here is where you make your pro or your anti-saccade. And we noticed a characteristic shape in the diameter of the pupil through our trial, that there was some baseline here that I've zeroed for now. And then the pupil starts to constrict a few hundred milliseconds after the fixation light comes on. This is a luminance increase, so it drives a constriction of the pupil. That's the pupil light reflex. What was intriguing is what we noticed late in the trial, before the target comes on, the pupil starts to dilate. And we were very interested in this dilation response because in the literature, we thought it was going to be related to cognitive control. And what we noticed was in the pro-saccade and anti-saccade responses, the constriction part of the response was identical because we matched the red and green colors for luminance. So they were isoluminant. And the difference here emerges because of the cognitive control late in the task. And we think that this difference in dilation on the pupil is due to the signals that I showed you previously that were in the superior colliculus that are different between pro-saccade and anti-saccade trials. So we think that's what's driving this difference. So now if we focus on this dilation being stronger on the anti-saccade trials than the pro-saccade trials, they're significantly different at this moment in time when the target comes on. So just ponder this for a moment. At this moment in time, before the target comes on, the pupil's telling us whether the subject's in a pro-saccade trial or an anti-saccade trial. And even better, Josh went on to show when these subjects made direction errors, the amount of this dilation was reduced so that it was more comparable to what we got on the pro-saccade trials. So in other words, we can measure at this moment in time on the pupil and get a readout of what's going to happen with anti-saccade behavior, which means the signals are being carried over to the other effector. So then Josh separated the distribution of saccades, the express saccades and the regular latency saccades, and he looked at this pupil dilation, and we got more dilation for express saccades than regular latency saccades, just like the neurophysiology we get in the monkey colliculus, significantly different. He took the anti-saccades and just divided them up into early anti-saccades and later anti-saccades. And again, we could get a significant difference in reaction time here, just like we got with the monkey neurophysiology. Okay, so now we can take this and go to these neurodegenerative patients and look at what was going on with their pupil responses. So this is the pupil responses in the pro-saccade trials, the anti-saccade trials. The gray line here are the control subjects. You can see all these neurodegeneration patients, all the cohorts we were studying, this attenuated constriction and abnormal dilation. You can see it even more dramatically here on the anti-saccade trials. The only normal dilation comes in this purple trace here, which is the ALS cohort that you'll remember I said was the most cognitively normal cohort. You can also see that the start of the pupil constriction up here has the same latency in all these neurodegenerative diseases, so that these differences we're getting, we do not believe are due to impairments on the retina. We have seen impairments in these early pupil responses, but we got them in a patient group with optic neuritis. And we might expect to see latency differences here in other early visual disorders, but we don't see them in these neurodegenerative diseases. Instead, all the actions later on when the cognitive control signals are kicking in. So again, the dashed line here are controls. The dark blue is mild cognitive impairment. The lighter blue is the Alzheimer's cohort. So you can see we can get this staircasing in the reduction of the pupil size. So we can look at the baseline measure that's been normalized here, but the baseline pupil size is reduced in Alzheimer's disease. It's reduced in MCI. We can measure the magnitude of the constriction response. It's reduced in Alzheimer's disease. It's reduced in MCI. We can measure the size of the dilation response out here. And again, it's reduced in Alzheimer's and reduced in MCI. We can do these same kind of games with the Parkinson's cohorts here. As we scaled with cognitive abnormality, it was really the dilation was the only one that was affected. We saw no difference with baseline, no difference with the constriction responses, but we get a dilation difference with the MCI in the demented Parkinson's group. I won't bother showing that. So basically, what I've shown you today is the first 5 rows here of different patient groups. Saccade behaviors abnormal, pupil behaviors normal or abnormal. I didn't have time to get into blink behaviors that are normal or abnormal, but so many of these putative biomarkers, we start to lose the forest for the trees. And so in the last couple of slides here, I'll show you the final approach we're using now. And this is being driven by our programmer, Don Bryan. And we're trying to build disease classifier models based on all these potential biomarkers that I've been showing you this morning. So we've got all these features like reaction time, error rates, amplitude of saccades, et cetera. In addition, we have time varying signals that vary through the tasks like the pupil, the blink rate. This is actually what the blink rate looks like. And so this yields 48 possible features that Don could use to build a classifier model, which is way too much data, but we can do a dimensionality reduction. And now we're getting into really complicated machine learning paradigms, et cetera here. But he can take these 48 parameters and reduce it down to a 2-dimensional space, where shown here now in red are the controls, in green are the PDs that are cognitively normal, in blue are the PDs with MCI, and purple are the PDs with dementia. And you get this idea of this trending from red to green to blue, where at least at the red and green stage, they're not differentiating very well. There's a lot of overlap. And so in his first classifier model that we could get 83% out of for the entire, or the area under the curve was actually 87% for the ability to discriminate the Parkinson's patients as a whole. When we looked at the cognitively normal Parkinson's patients, the area under the curve was only 83%. But for the mild cognitive impairment group, it jumps to 88%. And finally, for the demented group, it jumps to 94%. So his classifier is really good at pulling out these the cognitively impaired patients. And the bigger problem here with the classifier is not the patients, it's the control subjects, because at the moment of testing, they're controls. But if you're 75 or 85 years of age in your control, you are probably prodromal, pre-symptomatic for Alzheimer's or Parkinson's if we could just record from you long enough. Okay, I apologize for going so long, but I thought I hit 4 important points here, showing you how we could dive into using simple video-based eye tracking and our deep knowledge of circuits through the brain to show you how we could use measures of saccade and pupil to understand these brain circuits, and then how we could translate that to the human condition to study normal behavior across the lifespan and to identify biomarkers in various neurodegenerative and other diseases. And finally, how we can pull all of this together into disease classifier models, ultimately that could be there together in the clinic with an iPad, with a video-based eye tracker built in, that a clinician could be using to assist in their diagnosis. And finally, I'll just thank all my funding sources. I've had so many over the years. I run a big lab. I got a huge number of people that contributed to this. And individually along the way, I identified the key people who contributed to this talk. So thank you for your attention.
Thank you very much, Doug, for this absolutely fascinating and amazing presentation and work. I mean, I guess we will have, I have plenty of questions, and we already have many hands raised out there. So let's see if I have questions in the chat, and if not, I will just let it to my colleagues here or people who want to ask questions.
So I saw 2 hands up, but they went down, I guess.
Yeah. So Valerie has a question. Valerie.
Okay, thanks, Doug. I mean, I was doing a lot of thinking, especially the part about how this multimodal deep phenotyping of patients with neurodegenerative conditions. So I have a couple of other things, couple of questions. I was worried that your controls, like how do you know that they are actually going to develop disease, and does that affect your data? And you address that a little bit later. Did the eye tracking help when combined with the other measures to, in combination, give you a better prediction tool? So did it, if any of those things on their own probably might not be 100% predictive, or were they? And did the eye tracking improve the predictability?
Yeah, so I'll answer your second question first, because I can't really answer it fully yet. So in the different measurement platforms in our big Andre study, we are trying to build disease classifier models, like using speech and language data. We're using eye tracking data. And we fully anticipate that a classifier that's going to go across the platforms is ultimately going to be the best one. But we also want to include in that an analysis of what the cost is to the healthcare system. Like if an MRI can be used to help you with the diagnosis, but it's a $2,000 charge, and you can get the same answer with eye tracking for $30, society should go with eye tracking. And so we're trying to push that 2 ways. If I come back to your first question, which is the real stubborn one about the controls, we're doing this incredible recruitment to build this database with the goal of trying to follow the elderly people longitudinally. And our COVID moment came, as everybody who studied did anything in any lab. COVID severely attenuated our ability to recruit. We kind of made a conscious decision that we would focus all efforts on the last 2.5 years of just going after the critical patients in our midst.
Yeah.
And we put a pause on the control recruitment. We're hoping now, as things stabilize in Kingston and elsewhere, that we can go back to our controls, and we want to bring them back in again to see if the ones that were showing variability from a couple years ago, have they converted, or do they have a different diagnosis now? So it's a difficult question to answer. It really requires, you got to have a control cohort you're going to follow longitudinally.
And I deeply resent that you're considering people 55 and older elderly in your control group, but just saying.
Well, I'm taking the bullets myself. So if I were to show you the rudest birthday card my lab ever gave me, they were trying to plot the error rate in the anti-saccade task versus age, with a big red arrow of where I was on my 60th birthday and why I need to have like maybe power of attorney over the funds of my research account or something. And so, yeah, but it's sad what's happening.
All right. Caroline, you have a question?
Let's go to James had a question in the chat.
Yeah, James has a question in the chat. Okay. One of our students here at the Donald K. Johnson Eye Institute has a question. The question is, have you been in contact with the Canadian longitudinal study on aging at all?
I have not been in contact directly with them, but indirectly through our OBI study, the Andre study that I was showing, other elements on our team are now linking to the CLSA study that's ongoing. And so my understanding is it's mostly the people in the imaging platform, in the neuroinformatics, and a little bit of the wearable space. We are expanding into collaboration with the CLSA. But in the CLSA, since they didn't embrace eye tracking, I wouldn't say it's a dead end. It's an opportunity I see that's coming, but they don't have eye tracking data yet.
All right, thank you. Caroline.
Yeah, Doug, that was fantastic. It's very, I was really enamored by what you said about like the ALS data. Right, and so I think where some of it wasn't coming to significance, at least in some of the data that you showed. So one of the questions was with the eye tracking that you're doing across all the different conditions. I have kind of two questions. So one is like, does the change in significance for some of the diseases like ALS, where you think there's motor impairments, obviously, right, or at least certain aspects of the CNS will be impacted. Does it say something about? Does the eye circuits that you're looking at say something about what parts of the brain and circuits could be impacted when you see differences between the different?
Yeah. So, I mean, I could go on for a whole hour to answer your question. So I apologize. That didn't come out clearly in the talk at all.
Yeah.
So in ALS, the last movements that will remain that they can make are eye movements. Those are the last motor neurons to go. By the time the eye movements are affected, that's the end of the game in ALS.
Right.
So the actual motor apparatus in the brainstem is quite resilient to the pathology in ALS. But the pathology coming in the cortex is going to affect these descending circuits coming to the superior colliculus. So the cognitive impairments across all the neurodegenerative diseases, we think, are impairments in the frontal cortex and or basal ganglia that are impacting the signals coming to the brainstem. Not that there would be anything wrong in the brainstem per se. Okay. With the data I showed. So the anti-saccade task, pro-saccade task was really good at looking at cortical, basal ganglia, cognitive control. But it's not so good at assaying really low-level brainstem behavior. If I just go back to share my slide, and I'm hoping I got one slide here to make this point. And if not, I'll turn this off right away. Yeah. So I focused my whole talk today on the interleave prosaccade and anti-saccade task. We have this other task where we've got data from just as many participants, because this is also part of our battery, where basically you're just watching video clips for 10 minutes. You can do whatever you want. The only difference is what this effectively is like. It's like you watching TV and I'm standing behind you with the remote control, and every 3 or 4 seconds, I'm going to just change the channel.
Yeah.
So if you think about that for a second, that channel change is a giant visual perturbation, because every pixel on the computer screen is going to change. So we are going to send a giant visual transient into that circuit that I showed you way back at the start of my talk. And so that's going to have, as this visual transient runs through the brain, it's going to have implications on saccade behavior, blink behavior, pupil behavior. And this allows us to separate out a lot more of what's going on down in the brainstem now. So this task is a lot better for dissociating the circuits in the brainstem. And this is really exciting because now we're seeing the different proteinopathies are abnormal, but in different kind of ways. So for example, I showed you a little bit today on the pupil, where the pupil responses were attenuated in Alzheimer's disease, which is a tauopathy. But we have evidence the pupil responses start to get a bit exaggerated in Parkinson's disease and related synucleopathies. And with TDP43, it's not affected as well with the pupil. But then those same diseases are affected differently when we look at the blink. And so what we're looking at, I'm going to have to wave my hands here, but we've got neurodegeneration going on in the cortex, producing the cognitive impairments I showed. We're really excited because we've got neurodegeneration. Well, we're not excited about this from the patient's perspective, but there's neurodegeneration going on in the brainstem that's having small and subtle effects in the brainstem circuit that's almost you can't detect by the naked eye, but we can get with our biomarkers coming out of this task. So you're absolutely right. Some of the biomarkers are produced because there's damage in the cortex, but other biomarkers are produced because the damage was down in the brainstem. If I go back to my insane circuit, like the anti-saccade task was really good for the things up here on the upper left of this figure. The free viewing gets much better at dissociating this mess in the brainstem down here.
Very interesting. Thanks, Doug.
Well, this is in line, if I just can make a comment here, with what you see in the anti-saccade in children, because at that age, the whole inhibitory circuits from the prefrontal cortex are not developed yet.
Yeah, exactly.
So the phenotype that you see with the children is equivalent to the phenotype, I would say, that you see with elderly or neurodegenerative disease, because this top-down inhibitory control is weak or absent even in children.
Yeah, with the one caveat of the separation, I didn't do a good job of it in the talk here, but this separation of the early errors and the late errors and how that went on to these two different factors. And both of those factors are looking at executive control in the anti-saccade task. But one of them is this top-down inhibition to not look at the stimulus. That's the first thing. But then the brain has to let go of that top-down inhibition, because now you have to make a voluntary anti-saccade task, voluntary anti-saccade. So now the circuit becomes vulnerable to this competition between the automated command and the voluntary command. And that's a different mechanism in the brain that might involve some same areas and some same neurons, but it's playing out differently in time, that process. And that how, like, both of those processes mature dramatically up to 20 to 25 years of age. But then in normal aging, it's this factor 3 process that goes to hell, more so than just this top-down inhibition. So somehow, with all the damage going on in these neurodegenerative diseases, they've got enough cognitive reserve or whatever to still be able to exert at least some top-down control. And the flip side, if you go back to the younger children, on some trials, they can exert good cognitive control. They just can't do it 100% of the time, like a university student can.
Okay, thank you. Jamie has a question.
Thanks. It was a fantastic talk, really interesting. And I wanted to ask if you could comment a little bit more. You showed a little, it was wonderfully huge data sets, particularly with the neurodegenerative diseases. And you talked a little bit about comparing the different classifications, I guess, with regards to progression, at least with respect to, I think, Alzheimer's and Parkinson's. But mostly your talk kind of focused on this sort of classification and prognostic, I guess, aspect to using these biomarkers. And I guess I'm wondering if you could comment a little bit further about whether or not you think that or if you're looking at these subpopulations of diseases in more detail to look at the sensitivity, I guess, of these biomarkers with regards to progression, and whether there's sort of a more potential information, kind of like you did in terms of the aging analysis in the animals and in the healthy patients.
Yeah, thanks for that question, because it allows me to raise a really interesting response to that I didn't have time to put in the talk. And so absolutely, what we're ultimately interested in, what of these oculomotor biomarkers could be predicting disease onset, disease progression, outcome of some therapeutic, whatever. So to push that, you got to get to the patients that are asymptomatic. So how do you find them? So this is where you have to be opportunistic. So from the domain of Parkinson's disease, I'll bring up two very interesting cohorts we're studying. So one of them is a group of patients that have been identified with a LRRK2 gene mutation, that about 80% of them are going to convert to Parkinson's disease. And so this project's actually been led by Connie Morass and Tony Lang out of Toronto Western Hospital. And because they were part of Andri, and we have an eye tracker down in their clinic at the Movement Disorders Center at Toronto Western Hospital, we were able to get access to these patients that had the LRRK2 gene mutation and their families, so we could identify gene carriers who were asymptomatic, who were therefore prodromal, so that we tested them now 3 or 4 years ago. It was interesting that it was the pupil biomarkers that were sticking out with them, not the anti-saccade error rates. And now we have the ability to follow those patients now, those individuals in time, to see which ones will convert to Parkinson's. And then opportunistically, we've been able to do the same thing with a completely different cohort of patients that have been identified with REM sleep behavior disorder. So these are patients that early on are going to start to act out their dreams when they're sleeping, kicking and punching in bed. So that inhibition of the motor neurons is gone. A high percentage of the REM sleep behavior disorder patients will convert to some kind of Parkinsonian syndrome. Most of them convert to Parkinson's, about 60%. About 15% maybe convert to multisystem atrophy, and about 20% to Lewy body disease. And so again, I have a collaborator over in Germany who runs a very big sleep disorders clinic, sleep disorders together with movement disorders. So he gets all the REM sleep behavior disorder patients all over Germany get funneled through his clinic. So we do the exact same thing. We've got an eye tracker in his clinic. We're studying these patients to again identify which of these biomarkers are emerging before the clinician can say they have Parkinson's disease. And again, it's the pupil biomarkers. So it's suggesting to us that there's a key part of the neurodegeneration here in Parkinson's disease. And we know this from the Brack's work, that there is, in the different staging of the disease, there's pathology marching through the pons and the medulla. and it could be affecting locus coeruleus and other nuclei in the midbrain that are right around Edinger-Westphal and the central mesencephalic reticular formation, which is right where the pupil premotor circuit staging area is. And so as we look across those disorders, we start to see fascinating little things sticking up with the pupil. And so we think the pupil is going to be a key part of that early identification to help in early diagnosis, whereas some of the other measures I showed today will be good at following patients longitudinally. But we're really trying to focus efforts to go, I guess, upstream earlier in the lifespan to try to identify who are the individuals who are at risk.